be a mystery?
I believe it's still a mystery because the approach has been too crude for the complexity of what we're trying to solve. Interesting observation to make here, I think. The field began, you might say, of chemical evolution in 1953 with the publication of Stanley Miller and Harold Urey's paper. Well, really at this point, Stanley Miller's paper on the origin of life, showing how you could simulate in a laboratory glassware conditions that might have existed on the early Earth some 3 to 4 billions of years ago. In that same year that Stanley Miller showed how life might emerge from simple chemistry, Francis Crick and James Watson startled the world with the publication of their structure of the DNA molecule. So the whole genetic revolution, you might say, has begun in how complex we now understand increasingly life is, even at the simple, quote, simple level. It's far more complex than anyone ever dreamed at the time that the first model of chemical evolution as a way to explain how life began was presented. So you have the idea that first chemical evolution is going to show how the first cells began, and is increasingly, since that time, we found out level after level after level of inquiry that the simple cell is far, far from simple. And then it's just the pure complexity of the subject has shown that these crude blasting of chemicals technique with ultraviolet light or heat or any other type of energy source is just conceptually, it seems, increasingly the wrong approach.
Do you see a particular irony in the timing of Stanley Miller's experiments and the discovery of DNA by Watson and Crick?
Well, irony, yes, in the sense that one is trying to show how simple life is. The other one shows how utterly complex life is. And the two methods or approaches just don't seem to fit.
What are the major problems with origin of life simulation experiments?
Well, let's look at the prebiotic simulation experiments just for a minute, because you have to understand what is being done in these experiments. The goal is to simulate in a laboratory setting the conditions, as the theory says, of conditions that might have existed on an Earth 3 to 4 billion years ago. Now, in principle, there's nothing wrong with this approach. However, in order to get experiments that work, you have to constrain the conditions so that you can get results. Even the design of the original Miller experiment illustrates this. He arranged the flask in one way and no results. And you rearrange the glassware and you get results. So it was, why were the results, why do we consider the arrangement that gave results that set of conditions which match the early Earth instead of the other one? Well, it's because the one set of conditions, the glassware, gives results that matches the theory. Now, that's fine. Then you have to go on and consider, well, what else are you doing in this simulation experiment that corresponds with what you say existed on an early Earth?
Isn't it rather impressive that amino acids were produced in the Miller experiments?
Well, that's what was so impressive to many off the bat. You take a simple environment of methane, ammonia, water vapor, and hydrogen gas. No oxygen in these systems, by the way, because according to the chemical evolution hypothesis, oxygen would only appear on the scene after the first life began to produce oxygen. The first plant life would have produced oxygen. And then you can do these experiments without oxygen there, because you're dealing with it prior to the beginnings of life. At least that's the idea. It turns out there's a practical benefit of doing this. You don't get experiments to work if you have oxygen present, because oxygen is a poison preventing these type of chemical reactions. Another thing that is that if you use methane and ammonia, in other words, the so-called reducing environment, with some hydrogen gas present, you can get plenty of organic materials produced. And yes, it was very encouraging to the proponents of the theory when amino acids, a few amino acids, were discovered in the flask. In fact, over the years, more than a dozen such amino acids have been found in the Miller-type experiment.
How close is the development of amino acids to the threshold of life?
Well, you have to have many things besides amino acids. And if you only produce the suite of amino acids that are necessary for things, and there is a subset of 20 amino acids that we know in living things, you need those, but you need many other compounds and chemicals. But let's just deal with the proteins or the amino acids as their components first, because in these simple experiments, simulation experiments, you produce not only that set of amino acids, but many other amino acids. There are far more amino acids that are produced, and even in greater abundance than are produced of the 20 amino acids. And so you mix all those together with all of the other chemicals that are being produced, and you find out that the interfering cross reactions of these just prevents any type of meaningful buildup of chemicals that are sufficient to go on to the next step in making life. So it's a very difficult, almost intractable problem, I would say, chemically.
What are the steps involved in producing proteins from amino acids?
Well, the way typically these experiments are done, you do an experiment, you produce a glycine, an alanine, and a few other amino acids that we know exist in living systems, in true proteins, for example. And then you isolate those amino acids from the system, and you purify these amino acids, and then you use these amino acids separated, isolated from the myriad of other chemicals that would have been present in a, quote, prebiotic soup of chemicals. And then you react these amino acids together under other conditions to produce polymers. In other words, you're trying to build up, see if you can simulate the production of a protein.
Why are amino acids isolated during this process?
We want to separate the chemicals from each other in different stages of simulation experiments so that we can get a realistic handle on how the developmental process of chemical evolution occurs. If you don't do this, you have all the different chemicals mixed together, and they will mutually interact and react with each other. Now, why this is bad, or what happens is, amino acids will combine with amines or other groups that have say a carbonyl group, and you will get dead end process. You won't get meaningful substances.
What do you get?
Just a goo, just other substances that don't have any biological significance to them. And this is the really difficult part in trying to conceive how to do a simulation experiment. It's well known, nearly every one of these experimenters know that you can't do a realistic experiment according to what the theory says and get meaningful results. But if you just did that, everybody would get the same glop, and it's not a meaningful result. And so you have to have meaningful results to publish, and that's the reason why you separate things, so you can get have an idea of how you're going step by step to produce to go along the process of making life.
How can the investigator affect the outcome of a simulation experiment?
Well, you must have, in any kind of a simulation experiment, the role of the investigator. I mean, it's impossible to do experiments without the investigator. So when we wrote our chapter on the role of the investigator in these simulation experiments, we were trying to point out and make a distinction between those legitimate interferences in the experiment, such as stopping the experiment at a certain time and withdrawing sample for analysis. That's legitimate. You can do this. The experimenter can set up the laboratory conditions and set the parameters of the experiment, and that's fine. These are the kinds of investigator role that you would expect. What is illegitimate, and we felt like needed to be brought to the attention of students and other professors who were investigating this, and brought it out into the open, because, believe me, it's never brought out into the open. You can go to these international meetings, and there's two things that are almost never brought into discussion. One is the problem, as we've already mentioned, the role of these interfering cross reactions. If you discuss the role of the interfering cross reactions, there would be no other place for the conference to go. You would just focus right there. It would stop. There wouldn't be anywhere else to go, because you can't have any meaningful results. So nobody talks about it. That's frankly the way it is. The other thing is that besides the interfering cross reactions, you must not talk about in these conferences the role of the investigator to interfere in an illegitimate way with results, because if you did, it would become obvious that most of these experiments are unacceptable according to their own criteria for what we're doing as prebiotic simulation. That means, by the way, let me explain what I mean by that. According to the theory, we want to reproduce in a laboratory setting conditions that would have been on an early Earth 3 to 4 billion years ago, and that means no interference from God or any other outside forces, and either then or since. So we make a naturalistic assumption from the beginning to the present. Well, that means then that we cannot have, if we're going to simulate those conditions, we cannot have an investigator constraining the conditions that are outside the boundaries of what we've already said were acceptable for that prebiotic Earth.
How did you evaluate the different chemical evolution experiments?
Basically, we put the scale in such tentatively drawn. I mean, if we can show that it fits the parameters of a legitimate prebiotic experiment, then yes, by all means include it. And we'd be glad to move our line one way or the other according to how the data goes. But so far, the experiments are such that the investigator's role prejudices the outcome of the experiment in an illegitimate way. I just find it intolerable that men of science can allow this to go on and not make it vocal and not make it open, and certainly not, be more specific in their research papers. As I mentioned earlier, though, the reason why we don't do it, why it's not done, is that if you did, you wouldn't get meaningful results. And you can't publish a lack of results. You can't just do a research paper and say, well, I'll publish a paper of what I've done the last 2 months that hasn't worked. You've got to have some results. And it turns out that in 40 years of all these experiments, by and large, now there are a few exceptions, but by and large, most research papers have fit the category that we would call illegitimate, because the experimental parameters are constrained in such a way, the conditions are constrained in such a way that it would not fit a prebiotic Earth. And that's the primary problem that I see with these simulation experiments.
Are the initial conditions in the simulation experiments plausible?
Okay, let's look at the initial conditions of the early Earth. It started out first that the hypothesis was an atmosphere of methane, ammonia, water vapor, and hydrogen. You would have had hydrogen sulfide also, which you need for certain proteins and other substances that contain sulfur. But originally, the famous Miller experiment only contained methane, ammonia, water vapor, and hydrogen. Let's look at why methane and ammonia are in this experiment. It turns out thermodynamically, that is the energetics of the system that you're dealing with inside the flask, carbon that is in the reduced state, nitrogen that is in the reduced state, and that means the maximum amount of hydrogen connected to the atom, carbon or nitrogen. In those states, a reduced state, it's like having a molecule at the top of a hill. And what happens if you blast it with a spark or of electricity or ultraviolet light, or even heat it up to such an agitation point? Well, it's going to tip it over the edge of the hill, and it'll go downhill and bring about a chemical reaction. It's what is known as a chemically spontaneous process. So in this kind of an atmosphere with methane and ammonia in it, you can blast it with these energy sources, make the reaction go downhill, and bring about a production of amino acids. That's all straightforward chemistry. Fits the physics perfectly. Understandable. The really difficult part is trying to do this with carbon monoxide, or even more realistically, according to more recent investigations of what an early Earth would have looked like, carbon dioxide and nitrogen gas. Very, very poor results. If the early Earth was like the modern geochemistry picture of an early Earth containing carbon dioxide and nitrogen and some water vapor, it would be extremely difficult that this process ever worked. But what happens is, in order to even get things to work with carbon dioxide and nitrogen, put a little hydrogen in there. We need to have hydrogen. Why? Because when you blast it, you can still make the process work and get some products, but you must have the hydrogen present. This is a doubtful procedure too, but it does get around the problem of not having trying to deal with the problem of no methane or no ammonia on that early Earth, of which there is no geochemical evidence that it would have been there.
What did the Earth's early atmosphere contain?
Well, for example, the picture that geochemists give today is that look at the effluent gases of volcanoes would give you a pretty good handle on what what kind of gases would have been coming out of the early Earth, and it turns out to be carbon dioxide and nitrogen being produced.
Will the simulation experiments work with this atmosphere?
That's what I'm saying, that you get a very, very, very small amount of any organic compounds in this kind of environment. You must have a hydrogen there, and it really does help. And in fact, Stanley Miller has said on a number of occasions, well, I don't care what the geochemists say, you can't do the experiments without methane, so since we know that life is here, life must have had, these early Earth must have had methane on it. Now, this is the kind of reasoning that really dominates in certain quarters of origin of life studies. It's not the typical scientific approach that you would expect and you would like to see. It is dominated by a metaphysical condition, I would say. We know naturalistically that life is a result of these processes of chemistry. Well, how do we know this? Well, we're here. And so how can we get here? Well, we've got to have certain chemical reactions that will lead us to where we are. And what kind of conditions? Well, the only way we can make it work is if there was some methane and ammonia there. Therefore, they must have been present. So this must have been argument is used very often in this field, and it's very frustrating for those of us who are trying to move the field over to more realistic scientific approach to this problem.
There seems to be an underlying assumption that the origin of life resulted without any intelligent input whatsoever. Yet the simulation experiments appear to rely upon intelligent guidance. Could you comment on this irony?
Well, there's an interesting contradiction that comes in when you look at the stated assumptions about what the conditions on the early Earth were and what is required to make an experiment work. You have an early Earth, says the theory, void of any intelligent activity. That's by definition to do a naturalistic experiment. But in order to make the experiments work, the illegitimate role of the investigator, who is always intelligent, prejudices the direction or changes the direction over which it would normally go if that investigator did not do certain things. Now, let me give you a specific example here, because I think it's important for it to be understood how it works. The theory says that, for example, ultraviolet light would have been a major energy source on the early Earth. There's no reason to doubt that it wasn't irradiating the early Earth. So we want to simulate in our laboratory setting experiments using ultraviolet light. Now, in order to do the experiment, even with methane and ammonia present, you must use the shorter wavelengths of ultraviolet light, less than 2,000 angstrom units or wavelengths, in order to make the chemical reactions take place to give us a product. But what happens if you use the longer wavelengths, say greater than 3,000 angstrom units? What happens if you use those? If you use those, say greater than 2,000 angstrom units? Well, what happens is that the destructive processes of the products begins in very big time. And so you're destroying the products of this reaction thousands of times faster than you are producing the products. So, in a laboratory setting, if you use the full spectrum, for example, of course experimentally you cannot do this. We don't have a way to get the full spectrum, but we can produce longer wavelengths and shorter wavelengths. But these experiments are never done this way. We filter out the longer wavelengths, and I say that's illegitimate.
Are there any natural processes that would have filtered out destructive ultraviolet light?
If, in fact, there was a realistic filtering mechanism, then of course it would be an acceptable experiment. But no one has suggested any way, any possibility, or any mechanism for eliminating the longer wavelengths and just allowing the short wavelengths to react with the materials. And I think that, frankly, it's done because it gives the results that we have already specified as a stepwise procedure toward the development of living things. And interesting thing was this, another factor that must be considered, I think. By the time a body of criticism in the literature of Origin of Life was built up, okay, to show that these destructive processes would have far and away dominated the situation. By the time this literature was built up, the assumption was already set and permeated the whole field. We can do it. The monomer level, in other words, the first level of making amino acids, is presupposed nearly by everybody. And so the fact these criticisms are unheeded. And so we've gone on now to explain, well, how do we make proteins? The first 10 years of the field, say from 1953 until, say, 1965, 66, almost all, the vast majority of papers during that time was, how do we make amino acids? How do we make adenine, one of the bases in DNA and RNA? How do we make the sugars, the monomers, because you can't have a DNA or an RNA if you don't have sugars? Well, despite all of the body of criticism that says, well, it's not very realistic that you ever had a prebiotic soup that had adenine in it, or ribose in it, or that had very meaningful concentrations of these various amino acids from which you can make proteins, despite that criticism, the field today has presupposed that we have granite to stand on. And they don't even have sand to stand on. And they're trying to build polymers, proteins and nucleic acids, as if we have solid evidential basis that the precursor or components on which to build these polymers has already been established. And it has not been established. So the field now is dominated, majority of papers are dealing with the polymer level. So we're in a different decade and we're doing different things, but it hasn't worked out.
What additional steps would be required to develop life?
In order to have life, you have to have not only the amino acids from which you make proteins, and the nucleic acid, and the various components to make nucleic acids. But you have to have the the assembly for the cell structure, cell membrane. And the cell membrane is not simply a bunch of fatty acids that react and make a micellar or a bilayer of chemicals. It's so that you can have an enclosed boundary. What has been discovered in the last decade about the cell wall, the cell membrane, to show that it is utterly complex, extremely complex. It's perhaps one of the major discriminating factors in allowing certain materials in and out of the cell. Now, this structure must be there in order to have a viable living system. And no one has any clue how to do this. There are many things of which we don't know how to put together, how to assemble it. But conceptually, the problem has been for the last 25 years to make the monomers and then to see how to make the polymers, and then how to, and different research workers and groups are trying to work on different facets of the problem. But let's not kid ourselves. We're discovering more and more how utterly complex the living system is. And that's why the field is shifting and changing. We're either at the end of a paradigm where chemical evolutionary processes are being recognized as is the wrong approach. I mean, however much merit there has been seen in the past, we're rapidly approaching the age where more and more research workers are saying, well, we're going to have to try something else. We're going to have to do something else. Why? Because no results that are meaningful. There's not anybody in the world that can explain in a realistic way how to make a protein. There's not anybody in the world that can offer a realistic explanation for how to go step by step to make even a nucleic acid. And in a realistic environment now, with plausible conditions. And I have to put that qualifier in there, with plausible conditions, because people get Nobel Prizes for using constrained conditions, where you manipulate the conditions so that the chemistry only goes in one direction. But if you do this, that's no longer prebiotic. You see, that's the problem. We have to do it in such a way we can mimic a dumb, stupid nature, blind, chemical, physical processes. Now, if we are going to require something of the level of a Nobel Prize-winning scientist to manipulate the conditions, then you can do this with great difficulty to make some things, but still no one's ever made a bacterium. No one's ever made a cell of any kind. No one's ever made a virus, even. No one's ever made the kind of protein from just basic chemistry. Now, it can be simple chemical steps. Now, Fred Sanger and others have gotten Nobel Prizes for making, like, for example, insulin, but certainly this was not done with the kind of crude processes that the Origin of Life field is trying to use to mimic the process.
What is so difficult about making proteins or nucleic acids?
Well, the problem in trying to make a protein, for example, from even if you have just a pool of amino acids, is that the energetics is such that now it's an uphill process. Now, I mentioned before having methane and ammonia and blasting them with the energy and having it as a downhill process. Well, let's assume now all we have is a pool of amino acids that are the right set of amino acids to make a true protein. Well, how are we going to assemble the units, individual amino acids in a linear sequence to make a protein? In the first place, in a real protein, they're all left-handed amino acids, I guess you would call it. Except for glycine, which doesn't have any handedness. But all of the other 19 amino acids are left-handed amino acids. Well, when you do the simulation experiments, you get a 50-50 mixture. There's no way around that anyone has found. If someone could do a laboratory experiment and say that fit the conditions of something realistic on an early Earth where you just made left-handed amino acids, but this would be a tremendous encouragement to the field. In fact, that person would probably get a Nobel Prize. For example, if somebody can get a Nobel Prize for showing that, that an enzyme can have catalytic activity, then, which is a requirement. What did I say?
RNA.
Yeah, RNA is now recognized to have catalytic activity. It's not just a part of the nucleic acid or part of the informational system, but it is recognized to do the role of, on a somewhat more limited level, of an enzyme. So if this could happen, and is worthy of giving a Nobel Prize for this discovery, then certainly anyone who figured out a way to produce left-handed amino acids would probably be worthy of the same kind of thing, especially considering the amount of work that's already gone into it. Now that's one thing. The other problem is linking them together. In a real system, you can't do it without having an enzyme present. In fact, the assembly process it can't happen without enzymes. And the reason being, you must have the energy put at the right place to make the reaction go uphill. And that's the problem. It's a tremendous uphill. It'd be the equivalent of, I guess, being down in a valley and trying to go to the top of a mountain. And this doesn't happen without not only energy. If you put energy if you have gasoline, you have energy, but you have to have it in the right kind of engine. I mean, you don't just pour gasoline over your some kind of some kind of a wagon. You've got to have an internal combustion engine to use the gasoline to drive you up the hill. That's what the enzyme does. It uses the energy that you have available and couples it to the individual amino acids to bring about the production of the protein.
In addition to the energy problem in protein synthesis, isn't there a sequencing problem?
Absolutely. In order to have viable protein, you must have the right sequence. And the reason for that, according to the standard way of looking at it, is if you don't have the right sequence, the protein doesn't fold up properly to give its overall three-dimensional structure. Best way I can describe it is that the overall three-dimensional structure is said to be held within the sequence of the individual amino acids in the protein. If you had the right sequence, the molecule would fold up. Okay, I call this self-assembly. But you must get the right sequence. And you can't get the right sequence by just chemical processes that aren't directed. And they are directed in living system by the enzyme. But in a flask, they can be directed by the role of the investigator. So you can't get around the fact that either you have to have an enzyme or you have to have the role of the investigator. If you have the role of the investigator, that's an illegitimate entity for what you said is the prebiotic experiment. And that's the impasse this field has been in for some time.
History of life's origin, you refer to order, randomness, and specified complexity. Could you give us an overview of these concepts?
Yes, it's very important to make the distinction between order and complexity, and then between complexity and specified complexity, so I'll do that. A random situation is something that you can use, for example, with a sequence. Just pull letters out of a hat at random, and then the sequence would be a random sequence. This is what you would get if you do in the simulation experiments and you made a polymer. There might be some crude ordering. Some people are very impressed with the slight ordering tendencies that natural laws would give in sequencing these things. But of course, it's not like order that you have in a crystal. So an order would be like A, B, A, B, A, B, A, B. This would be a sequence of order. And a random sequence would be like A, J, B, Q, T, you know, something like that, where each letter, you might have a run of 3 or 4 that are consecutive A, but in the long run, each one has equal or near equal probability of appearance out of the sequence. Then a specified sequence is one that has like a written message, T, H, E, D, O, G, you know, the dog, and then spell out in the sequence, the dog ran. This is a sentence that's spelled out, fits the vocabulary, fits the grammar, and the proper syntax. It is this kind of distinction that you can recognize as a way for order, and then randomness, and then specified complexity. Now the thing is that in the examples are that the results of stochastic chemistry, what happens in a lab, in a flask, is an example of a sequence that would be ordered. Would be random. Complex is a random state. Order would be like a crystal. Everything is specified in a particular sequence, a defined arrangement. But specified complexity, it's complex. Some have said this. It's like a random situation where it has certain rules that are like language rules. So it's not a totally ordered arrangement like A, B, A, B, A, B, but it fits the characteristics of a language. And we see that in a language system, but the same thing happens with the DNA and protein. It also happens with making, for example, making a computer, or anything, artifacts, or other examples of specified complexity. Mount Rushmore would be an example of specified complexity. In fact, you don't find examples of specified complexity except the structures of artifacts that people make, and DNA, protein as examples.
What do you think the presence of specified complexity in a living system indicates about its origin?
In terms of the origin of specified complexity in living systems, I would say that if we had any examples available that unrestrained or unconstrained natural processes produced specified complexity, then I would be prepared to go along with the community of scientists that are saying that natural processes produce living systems. But we don't have anything like that. Every single example of specified complexity we have is either an artifact produced by a human being, or else it is the result of the living system. And so, like the structures of DNA and protein fit the pattern of what human beings produce. One way you can look at it, it's easy to understand why for so long people have used the analogy that a human being or a living system is like a machine. The machine analogy to living systems has had a long record now, and it's been very productive in a lot of things that have been accomplished in scientific approaches. But the machine never arrived at by spontaneous equilibration of chemistry and physics. These forces must have certain boundary conditions in which they operate. And this is what human beings do to make artifacts. We can make machines ourselves, but we don't find machines arising by natural processes. The same situation. So I would say that really we haven't progressed much in our understanding about this question. So I still conclude that the data supports the position that specified complexity is somehow, whether we like it or not, the result of intelligence.
In inferring the necessity of intelligence to produce life, haven't you ventured from the realm of science to religion?
Oh, I know that everybody says this is the venture of religion, but you simply look at the structure of what you're dealing with. It fits the pattern of experience has taught us only results by intelligence. All it takes to show that this is not the right answer is to find a single example of a natural process that produces specified complexity. And then we would have to really rethink things. For example, it's like the example of William Paley is in the watch is classic. Everyone has heard about this. You find a watch and you know that someone with intelligence has been there and made the watch. Well, this is recognized, of course. Now there's an argument that is often given to explain this in the chemical evolution crowd and literature. And it goes like this. Well, we know that it takes intelligence by way of someone to make the watch. If we found a watch, we would know that. But this is inorganic materials. We're dealing with a living system, which, and then the fact that we have to have human beings that manipulate the conditions to make these experiments work and publish them as prebiotic chemistry experiments. Isn't this a violation of the rule of naturalistic processes? Well, no. Why not? Well, because natural selection produced the investigators. It's a circular argument again that I'm trying to address earlier about we know that the early Earth must have had methane on it. Why? Well, we can't get the experiments to work without methane. This is no help. This is not a scientific approach. This is pure philosophy. And so, yes, I would say that my view is not only empirical and fits the data, but it fits our experience, that intelligence is the direction we ought to be looking for accounting for the information in the living system.
Could you summarize the reasons why you believe intelligence was involved in the origin of life?
The overall complexity. It's such an inordinate level of complexity, plus it fits the pattern of my experience. We don't find anything like this, the result of natural means and processes. It's only the circular argument of saying that we are here by the result of natural selection, which produces, in other words, natural selection produces us, we produce artifacts. Therefore, we don't have to worry about the fact that the watch is produced by an individual, and so we don't have to worry about the successful experiments because we find that human intelligence was produced by natural selection. Well, you go around and around in a circle. The fact is, we have such high-level complexity. Where do we find it? In living systems. Now, we have to have some way to break out of the circle, it seems to me. Now, you either have to rule this answer by religion or some other philosophy, and I say that by and large, what is being happened right now by the failure to follow experience, we are demanding that our students and our professors only look at natural process, and it doesn't fit our experience, and I think every researcher in the world knows it. It knows that it fits the pattern of Mount Rushmore and a watch. This is not the issue. The issue is it's unthinkable it could be anything else but natural process. Why? Well, it must be natural process. This is philosophy.
How is the mystery of life's origin received by the scientific community?
Well, I think when the book came out, of course, we were a bit apprehensive. In 1984, when the mystery of life's origin hit the world, we might say, there were very few people who were acknowledging the kinds of things we were saying. Well, in 9 years, or then a decade since then, there's been a tremendous shift. The kinds of things that we've talked about, and that's in this book, is generally regarded more and more favorably among the researchers. They know that the chemistry is not favorable to the production of living systems. It doesn't go that way. But it's standard textbook orthodoxy nonetheless. But in terms of going to the Origin of Life meetings, going to and interacting with the researchers who do the work, they're very candid. They're very upfront, and they know what's going on.
What are the major objections to your current point of view?
They say you can't do research if you hold design. It's really a strange kind of logic that works. We're going to hold a theory that natural processes produce this and continue to look for natural ways to explain it. And by the way, I want to put a parenthesis in here. I'm not at all suggesting that we should stop research on natural process approaches to the origin of life. I'm not at all suggesting this. In fact, I think it would be a huge mistake if we did stop that. But I am saying that along while we pursue natural process approach to the origin of life, it ought to be that the field should be wide enough to entertain other possibilities, other approaches. If these don't work, let's look at some others and maybe continue to work this. Maybe one of these will work. But it's sort of like the idea that if you say that only natural processes are at work, it's sort of like taking your geology students to Mount Rushmore and saying, look at this mountain. Come up with some wind and erosional process or other set of natural factors that would account for these faces. I'm saying that we have to be guided by the structure of the effects we see and let that determine the course of our inquiry, not the other way around.
What was Dean Kenyon's response to your critiques of his book, Biochemical Predestination?
Well, the first contact that I had with Professor Kenyon was a letter after having read his book, and a few years later having done my own original response to it, and I contacted him with an essay that I had written. It wasn't the manuscript for the book, it was an original essay that I had written prior to that. And he wrote back, was very positive to the criticisms that I had given, and suggested that he too had changed his mind about a lot of this and encouraged me to continue. I think that his shift had already happened before I contacted him, but certainly he was a big encouragement to me in terms of continuing along. You can imagine what it was like. I felt like myself, I was virtually alone out there. There may have been other critics who were saying these things, and obviously Kenyon was, but I hadn't read any of it. And I couldn't find anything in print that supported the views that I was coming to. So I was very tentatively putting forth, sort of like a child going to the ocean, never been to the ocean before, and you're afraid of this. You don't know whether you want to get out there and venture forth or not. And that's frankly the way I felt about the opinions I was coming to about the origin of life field and evaluation of the experiments. So when we presented the essay to Kenyon, and he was favorable to that, I began to correspond with other people with one of the drafts of this book. And we heard from a variety of different people who were doing research in the field, and they gave very valuable feedback. And what I really expected to find was an avalanche of resistance and criticism. And do you know that we did not find it? We found that, by and large, the chemists agreed with the chemistry statements, the geologists agreed with the geology statements, and the engineers or the physicists agreed with the physics statements. But here's what I call the Swiss cheese mentality, or the Swiss cheese view of knowledge or science coming into play here. Someone who was an expert in chemistry would see what we had done there and would say, well, yes, that's fine, but your problems are over here in geology or physics or something, or biochemistry. And then someone else who was expert in geology or physics would read those parts and would say, well, those parts are fine, but your chemistry needs to be worked on over here. And what we've discovered was that people's knowledge in their own area was up to date, but their knowledge outside their discipline was was behind a few years, and they hadn't realized that enough criticism over the whole field had been such as to basically question the credibility of this whole enterprise as the dominating paradigm for how life occurred on this planet.
What was his response to the mystery of life's origin?
That was an interesting experience. Let me tell about the time that I first met Professor Kenyon and the response that he gave to the book. I had wanted him to write the foreword to the book because his own book had stimulated my own thinking so much in the beginning of it. And I went to California in San Francisco State and met him after he had already had the manuscript for some time. And went in to see him, and he was very, you know, no clues given with body language or gestures or smiles or anything like this that he accepted the point of view of the book. And I was really somewhat apprehensive that how he might have found all manner of objections that would just undermine the whole thing. Because at that point, as I mentioned, we still felt fairly alone out there and stranded and isolated. And after a few minutes of just quiet in his office and discussion about general things, I said, well, Professor Kenyon, what's your response to the book? And he was just became very animated after a few seconds. He says, I really like it. And I don't know what caused me to say this, and my first response was, then why don't you write the foreword to the book? And as I can remember, he said, well, I was hoping you would ask me that. And so that's how he became to write the foreword. But he was very favorably impressed with the overall treatment. He had some critical comments to make here and there, and we addressed those. But I think that he felt like it was the kind of response that he wanted to give, but because of administrative duties, other things, didn't have time to go back and make those changes. I hope he will revise and update his own book, because it was certainly a masterfully done work. There's nothing wrong with this approach. However, in order to get experiments that work, you have to constrain the conditions so that you can get results. Even the design of the original Miller experiment illustrates this. He arranged the flask in one way and no results. And you rearrange the glassware and you get results. So it was, why were the results, why do we consider the arrangement that gave results that set of conditions which match the early Earth instead of the other one? Well, it's because the one set of conditions, the glassware, gives results that matches the theory. Now, that's fine. Then you have to go on and consider, well, what else are you doing in this simulation experiment that corresponds with what you say existed on an early Earth?
Isn't it rather impressive that amino acids were produced in the Miller experiments?
Well, that's what was so impressive to many off the bat. You take a simple environment of methane, ammonia, water vapor, and hydrogen gas. No oxygen in these systems, by the way, because according to the chemical evolution hypothesis, oxygen would only appear on the scene after the first life began to produce oxygen. The first plant life would have produced oxygen.
Be a mystery.
I believe it's still a mystery because the approach has been too crude for the complexity of what we're trying to solve. Interesting observation to make here, I think. The field began, you might say, of chemical evolution in 1953 with the publication of Stanley Miller and Harold Urey's paper. Well, really at this point, Stanley Miller's paper on the origin of life, showing how you could simulate in a laboratory glassware conditions that might have existed on the early Earth some 3 to 4 billions of years ago. In that same year that Stanley Miller showed how life might emerge from simple chemistry, Francis Crick and James Watson startled the world with the publication of their structure of the DNA molecule. So the whole genetic revolution, you might say, has begun in how complex we now understand increasingly life is, even at the simple, quote, simple level. It's far more complex than anyone ever dreamed at the time that the first model of chemical evolution as a way to explain how life begins are the amino acids as their components first, because in these simple experiments, simulation experiments, you produce not only that set of amino acids, but many other amino acids. There are far more amino acids that are produced, and even in greater abundance than are produced of the 20 amino acids. And so you mix all those together with all of the other chemicals that are being produced, and you find out that the interfering cross reactions of these just prevents any type of meaningful buildup of chemicals that are sufficient to go on to the next step in making life. So it's a very difficult, almost intractable problem, I would say, chemically.
What are the steps involved in producing proteins from amino acids?
Well, the way typically these experiments are done, you do an experiment, you produce a glycine, an alanine, and a few other amino acids that we know exist in living systems, in true proteins, for example. And then you isolate those amino acids from the system, and you purify these amino acids, and then you use these amino acids separated, isolated from the myriad of other chemicals that would have been present. And then you can do these experiments without oxygen there, because you're dealing with it prior to the beginnings of life. At least that's the idea. It turns out there's a practical benefit of doing this. You don't get experiments to work if you have oxygen present, because oxygen is a poison preventing these type of chemical reactions. Another thing is that if you use methane and ammonia, in other words, the so-called reducing environment, with some hydrogen gas present, you can get plenty of organic materials produced. And yes, it was very encouraging to the proponents of the theory when amino acids, a few amino acids, were discovered in the flask. In fact, over the years, more than a dozen such amino acids have been found in the Miller-type experiment.
How close is the development of amino acids to the threshold of life?
Well, you have to have many things besides amino acids. And if you only produce the suite of amino acids that are necessary for things, and there is a subset of 20 amino acids that we know in living things, you need those, but you need many other compounds and chemicals. But let's just deal with the protein was presented. So you had the idea that first chemical evolution is going to show how the first cells began, and is increasingly, since that time, we found out level after level after level of inquiry that the simple cell is far, far from simple. And then it's just the pure complexity of the subject has shown that these crude blasting of chemicals technique with ultraviolet light or heat or any other type of energy source is just conceptually, it seems, increasingly the wrong approach.
Do you see a particular irony in the timing of Stanley Miller's experiments and the discovery of DNA by Watson and Crick?
Well, irony, yes, in the sense that one is trying to show how simple life is. The other one shows how utterly complex life is. and the two methods or approaches just don't seem to fit.
What are the major problems with origin of life simulation experiments?
Well, let's look at the prebiotic simulation experiments just for a minute, because you have to understand what is being done in these experiments. The goal is to simulate in a laboratory setting the conditions, as the theory says, of conditions that might have existed on an Earth 3 to 4 billion years ago. Now, in principle,